Everybody. I am Karina Yeager, and I am really happy to introduce to you Dr. Assaf Anyamba. He is a senior research scientist at NASA Goddard, and with the Goddard Earth Sciences Technology and Research at the University of Maryland, Baltimore County. He did his bachelor's in geography and economics from Kenyatta University in Nairobi, his master's in geography from Ohio University in Athens, PhD in geography from Clark University, and his postdoc at NASA Goddard. He specializes in the utilization of time series satellite-derived vegetation measurements to study interannual climate variability and applications in global agricultural monitoring and vector-borne diseases. And he grew up in Kenya, in the Western Highlands of Kenya, herding sheep and cows. And I am very happy to introduce him today, and we're looking forward to your talk. Thank you.
Can everybody hear me?
Yes.
Okay.
It actually works really well.
Huh?
Yeah, it's
Okay.
really good.
So, yeah, thank you very much, Karina, for that introduction, and to the entire seminar committee for inviting me here. It is a good opportunity to visit Long Island for the first time. I've never been to this part of the country, and I'm very much pleased to be here today. So for the talk today, I'm going to talk about global El Niño-Southern Oscillation teleconnections, a topic that I think many of you in the marine and atmospheric sciences know, so I don't have to go into a lot of explanations. And then the second part is how this phenomena impacts vector-borne diseases globally. And then I'm going to have one example of that that is applied at continental level. So my talk in terms of outline is going to go like this. I'll provide some background and also some data sources for disease modeling, how we treat these data sets, and then talk briefly about ENSO Teleconnections and how we link that to the biology of diseases. And then I give an example of Rift Valley fever and developing an early warning system for this particular disease. So on this slide here, you can see a number of diseases that are given here. This is drawn from actually broadcast by NPR in 2023. And it provides there a list of diseases that are of importance biothreats globally. These include Nipah virus, Congo Crimean fever, Lassa fever, Rift Valley fever, which I'm going to talk about, Zika, Ebola and Marburg, Middle East respiratory syndrome, MERS, SARS, severe acute respiratory syndrome, and then disease X. I would say disease X, we actually experienced disease X during the COVID pandemic. So this collection of diseases are actually similar to what is listed as serious biothreats by various government organizations, including CDC, DOD, and USDA. Now, why is it important to study these diseases? As you will note, some of them like Rift Valley fever, Zika, Ebola, are related to what is going on in terms of the environment. And they affect various factors to do with vectors that spread these viruses. For example, if you look at temperatures, it has an influence of development rate, larval survival, and mortality. If you look at humidity, it has an impact on the biting rates of the vectors or vector competence. Competence is basically the ability or how effective the vectors are in terms of transmission. And then if you look at rainfall, it has an influence of the breeding habitats, say collection of water bodies where the vectors can breed, and then also food availability, like you think about rodents. So all these factors have an influence of population dynamics of vectors and disease transmission. Now, even though we look at those factors, they are what I will call X factors in terms of disease transmission. And these are in terms of travel and trade. And we have seen it, especially with COVID, that everybody can relate to. And the recent disease outbreaks that have been influenced by transport, you can think about it as West Nile virus. Before West Nile virus was not in the United States. It got planted first here in New York, and then within a year, it has spread throughout the country. Chikungunya virus, if many of you remember, during the period of 2015, 2016, there was a big outbreak in South America of chikungunya, dengue, and Zika. And we had a number of imported cases in the US, especially in Florida. And then, of course, the classic example is COVID, where in this particular case, the human was actually the vector. So we are capable of being vectors of disease. So if you think about it in terms of transportation, especially air transport, we have thousands of flights every year, every day going on around the world. And then also, if you think about trade, this is a big thing right now, trade. Through merchant ships, they are capable also of actually spreading pathogens and diseases from region to region. Now, as I mentioned before, we can derive all these data sets that are background drivers of particular vector-borne disease outbreaks. So we can obtain information on temperature, humidity, rainfall, and vegetation from remote sensing measurements. The importance of remote sensing measurements is actually the global coverage of it, the sampling frequency, and then also by creating a baseline of this data, we are able to make relationships between the phenology of the vectors and the outcomes in terms of disease outbreaks. So in doing so, we can actually utilize a platform of remote sensing to culture or design models that can be used in terms of risk assessment or predicting areas where diseases will occur, and therefore being able to undertake measures either to minimize or prevent these diseases from happening. And looking at satellite measurements, there are various data sets available for this. We have a collection of about 40 to 42 years of global sea surface temperature measurements, precipitation at the same time length, land surface temperature from MODIS, about 21 years, and vegetation measurements, which you can think about it as defining habitat suitability. That data is available now at global scale for 40 years. All these data sets, global data sets, are at various resolutions. With vegetation, continuous vegetation measurements, now we can get that data from MODIS at 250-meter resolution. Now, the data that I have been looking at mainly is the normalized different vegetation index. As Karina mentioned, Karina and I used to work in a laboratory at Goddard that was the first to create a global mosaic of this vegetation metric called normalized vegetation index measurements. And it's basically exploitation of measurements in the visible portion of the electromagnetic spectrum and the infrared. So by looking at measurements in those two portions of the spectrum, you can derive this measure that we call vegetation index. There are various types of vegetation indices, but the most common and widely used is NDVI. And as you can see here on the graphic in the bottom right corner, this is comparing NDVI or vegetation index with rainfall. And you can see that there is a good correlation of that over this semi-arid area of Africa called Sahel Sudan, up to a limit of about 900 millimeters of rainfall. So if you compare NDVI measurements during the growing season, which is typically June to October, and rainfall at the same time period from rain gauge measurements, you get a near perfect correlation between the two. Why this is important is that for most regions of the world, and I would say in the Southern Hemisphere, over time, there is a reduction in the number of surface meteorological measurements. So what has happened is that over time, these kind of measurements have become a proxy for studying what is going on in the environment because of the high frequency of the satellite measurements, daily to 10-day to monthly, and therefore being able to capture the dynamics in terms of what is going on the land surface over time with these measurements and relating them to various aspects of the environment, including diseases, including agriculture. But the important of this is that having data is not good enough. You have to get valuable information from it. And I'm sure this is something that all of you who are in SoMA understand. If I give you just measurements for today and tomorrow, it's some information, but it's not very valuable information. So from these measurements, the important thing is to calculate anomalies. And you can see these two sets of graphic here. These graphics are called Humbola. When I talk to many people who are not coming from a climate background or a meteorology background, they don't know what Humbolas are. But I think this is one of the most important inventions in meteorological climate sciences because it enables you to look at space and time within the same frame. So what you see here on the left is NDVI measurements at over a slice of region in East Africa between 1 degree north and 5 degrees north, summarized between 33 degrees east and 44. And then on the right is the corresponding land surface temperature. Now, all the areas that are shown in brown are areas of below normal vegetation conditions or the ones associated with drought periods. The greener periods are associated with abundant rainfall and therefore positive departures. If you look at land surface temperature, what you see, the areas in blue are when the land surface is cooler than normal. And then in yellows and red will be those periods that are associated with the drier than normal conditions or drought. Two periods I want to point out there is the period is in 2006, 2007, where you have greener than normal conditions and lower than normal land surface temperatures. And then the period of 2005, 2006, where you have below normal conditions in terms of vegetation and above normal temperatures. These are signature drought events in these particular regions. And these are the kind of measurements or information we can then use to drive to drive different models, either looking at models of agriculture or looking at models of specific vector-borne diseases. Now, these aspects of El Niño-Southern Oscillation, I think many of you know it. It is part of the natural mechanisms of interannual variability of the climate. And in some of the areas where Karina has been working in South America, reconstructions of El Nino go up to 500 years when you look at different ice cores in the mountains of South America. The important thing is because this is, I would say, quasi-regular repeating patterns where you have conditions that are cooler than normal in the Eastern Pacific during particular periods, and then you have warmer conditions generating a cycle between 6 and 7 years. The important thing is, even though the variations are only in the Central to Eastern Pacific, because of the sheer size of the Pacific Ocean, any kind of perturbations in sea surface temperatures in this region has global consequences. These are global consequences that are then called teleconnections. So you will find that during the El Nino events, you have wetter than normal conditions in Eastern Africa, in a large part of the Eastern Pacific, in the southern US, and also in the area of Argentina and Uruguay. And then on the other hand, you have drier conditions over the Sahel, over Southern Africa, over Southeast Asia, and in northeastern Brazil or North and South America. When the system switches over to a La Nina, and we have just been going through a La Nina period that is ending right now, the manifestation of that is wetter than normal conditions in areas that were drier during the El Nino event, and drier conditions in areas that were wetter during El Nino event. So you can see that this pattern is part of the internal climate mechanism and is repeating. It is 6 to 7 years, though it's not clear that it's going to occur with that frequency. That you have periods like in 2001, I believe, no, the period between 1991 and 1995, where you almost had a continuous El Nino every year. So this is something that we have to monitor carefully because of the consequences it has on different aspects of agriculture and disease outbreaks. So what we did during the outbreak, during the El Nino of 2015-16, because everybody had been talking about, okay, El Nino this, El Nino that, disease outbreak here, disease outbreak here, but nobody had ever put a full picture together of actually what happens about during an El Nino event. So what we did was to compile data during that outbreak period between April 2015 and April 2016. We use this particular system known as PROMED that they collect outbreaks of different diseases worldwide. And then we had students, intern students, collecting this data every week during that particular period for 1 year, and then being able to put this picture together. So the picture that then ended up happening is shown on this map here on the right, and you can see a cluster of cholera outbreaks over in East Africa, shown in blue, you can see another cluster of dengue in Southeast Asia. And then what I talked about in South America, you can see that these multiple outbreaks of dengue, Zika, and chikungunya, during that particular period. Also, you see in North America, the clusters of hantavirus occurring in the area of Western US, covering Arizona, New Mexico, and Colorado. So there is a tendency of these disease outbreaks to occur when an El Nino occurs. And the important thing is to relate them to the background climate. So for example, when you see an increase in cholera cases is typically associated with wetter than normal conditions during an El Nino event. On the other hand, what we have established from doing this kind of studies is that dengue, Zika, and chikungunya will occur more often during drier than normal conditions. And then this brings about questions because most people think that to have these vector-borne diseases happening, it should be raining. Now, this tells us that different vectors are adapted to different conditions. So the vectors of Rift Valley fever are adapted to wetter than normal conditions. The vectors of dengue and Zika are adapted to drier than normal conditions. So from this picture here, what we found out is that there are particular situations where having a forcing, for example, above normal rainfall due to during an El Nino event will result or would trigger particular vectors, for example, Rift Valley fever. And then on the other hand, in some cases, like for chikungunya and dengue, it will be an amplifier. The difference there is that some of these diseases, like for example, Zika and dengue, are always circulating in the environment. But when you have this amplification due to dry and hotter than normal conditions, you then increase the chance of having an outbreak. And these two graphs here illustrate what happened during the period 2015-16. On the left is a representation of cholera in East Africa, and you can see the peak period between December and March 2016, basically representing what I had shown in the map. And then if you look on the right, you have chikungunya in the Americas, and you see also the amplification of the outbreak during the 2015-16 period. I'm sure that many people didn't understand this graphic until COVID, but this is typically how diseases evolve in a situation where you don't have control. So we had this idea of flattening the curve. These particular plots don't show any flattening because there is no treatment, there is no building up of immunity in the population. And what you are seeing is that the disease is spreading in the environment over time. And then as the population builds immunity to that particular disease, like if you think about for chikungunya, you can see that there are less and less number of cases through time compared to 2015 and 2020. So the next example that I'm going to talk about in detail is this disease called Rift Valley fever. And I'm sure most of you have never heard of this disease, but it's important, an important biothreat disease. The US government took a lot of interest in this particular disease due to the outbreak of Rift Valley fever in Egypt in 1979 or 77 thereabouts. The other important thing is a hydrologically driven vector-borne disease. So it has very much to do with rainfall and what is happening on the land surface. The other thing is a high consequence disease, especially for livestock. Mortality rates of 80 to 100%, especially in sheep and cattle. Human, if you look at literature, it's mentioned that 1% of humans infected with Rift Valley die, but we have seen as much as 25% of humans that are infected with Rift Valley fever die. If you think about the case of Sudan in 2007. The other thing is the consequences to trade. Many of these countries that are impacted by Rift Valley fever trade in livestock. They sell livestock to other regions. For example, there's a lot of livestock going on, going from Eastern Africa, Southern Africa into the area of the Middle East. They're also exporting beef and other livestock products to other regions of the world. So you can see it has having a very big economic impact in terms of trade. Also, you can consider in terms of nutrition. When outbreaks happen, people are not going to consume a lot of protein from livestock. USDA estimates that if Rift Valley fever were ever to be introduced in the US, the impact will be about $1 billion. But if you think about it in terms of our livestock industry, the impact is estimated to be between $8 and $8 billion, $80 billion. So if you think about the stock value of companies such as McDonald's under these conditions, and I'm sure you see what is going on, you can see that it will have a very big impact on the economy. So if you think about it as well, I mentioned you have impacts on livestock. You also have impacts on human beings. So this particular disease is a very important pathogen in terms of being crossover. You hear a lot about One Health is a perfect example of a One Health disease system because it impacts both humans and animals. And therefore, in the past, some particular countries, especially the old Soviet Union, tried to develop this as a bioweapon. In the 60s, this was experimented in the old Soviet Union in developing Rift Valley fever to be a potential bioweapon. Now you can say a bioweapon, how that manifests itself. Say, for example, tomorrow morning we wake up and you tell me that there is an outbreak of Rift Valley fever in Helsinki, Finland, or in Ottawa, Canada. Those are areas that are not, oops.
Whoa. Oh, I think you just, the middle button
Middle button?
or the top button. There's like a,
Sorry about that.
oh, you're good. There you go. Okay, you should be back in business.
Yeah, so if you hear that, Rift Valley fever outbreak is happening in Helsinki, Finland, or in Ottawa, Canada, of course, those are areas that are not endemic to Rift. So it's likely to be a potential biological attack in those particular areas. So it's very important to understand the patterns and dynamics and the frequency of these events in endemic areas, mainly in Africa and potentially in the Middle East from the year 2000. However, in order to understand that, you actually have to understand the biology of the vectors and the environment in which the vectors of the disease prosper. So in this graphic here is a representation of what is called an endemic circle. Endemic circle is a situation where the disease is just circulating in the environment. where there is sporadic seeding every year, and then the disease is occurring at a very low level. So that would be, for example, during the seasonal cycle, there is rain, the vectors emerge from the environment. The rain is not so much, but these particular vectors are emerging, spread the disease at a very low level, and then you don't have a situation whereby an epidemic is developing. That is one theory. Okay, this is not foolproof, especially given the recent studies we have been carrying out. And then you have this particular period, which is the epidemic period, when you have large-scale outbreaks. And these periods are often exemplified with above normal rainfall, like during an El Nino event. So during this particular period, large areas of the landscape flood, and there are many production. There is a lot of production of the vectors in different regions. And then what happens is you have millions of these vectors that emerge from these habitats. And what happens is they spread the disease first to livestock and then to human beings. And if you think about it, if the rain continues, say for a period of 3 months to 5 months, like will occur during an El Nino, you are generating a large number or a large population of vectors that are potentially going to spread the disease. So satellite vegetation measurements are able to give us an idea of monitoring these situations over time because we have the time series collection of these data. Now, in order to understand actually what happens with vectors in particular environments, the earliest work that was done in terms of the relation between rainfall and vectors was carried out by Dr. Ken Linthicum, ex-Army and ex-USDA, who introduced me to this work. These graphics here show you on the top left is in blue color. The blue color bars are the rainfall, daily rainfall during October to January 1982-83. The graphic that is in red show you the water height in a dambo. Dambo is a shallow depression in the ground where flood water will often collect. And then the graphic at the bottom is showing you the number of vectors that were harvested from that particular dambo. Now, the important aspect to note here on the bottom graphic is that between 5 and 10 or 15 days of flooding this area, you have an emergence of what are known as Aedes species of vectors. And then as the area continued to be flooded, you had a large number of emergence of Culex species of vectors. Knowing these vectors is very important. The Aedes species are the primary vectors of disease. They are the ones that carry the virus. So they are the primary vectors. They spread the disease to the livestock first. And then the next generation, which is the Culex, that are also in large numbers, are the ones that are very efficient, are spreading the disease further to livestock and the human beings. The interesting thing about this study was actually carried out during a very dry period. So this was based on artificially flooding this dambo and then being able to harvest different vectors from that dambo over time. So if you think about it, during an El Nino event, think about an area the size of Texas, New Mexico, Arizona. you think about that whole area being flooded for a period of 3 to 4 months. And then you are talking about that region having habitats that are shown in the bottom right corner, which are known as dambos. So there are many of these particular structures on the landscape. So you are talking about generating millions and millions of these vectors that potentially carry the disease. And you are then talking about that whole area being affected by an outbreak of Rift Valley fever. So the important thing is actually, in terms of disease, understanding the vector dynamics and different conditions in which they emerge. So this was based on artificial flooding, and it is not until 34 years later that we actually repeated this under natural flooding conditions. So this is from a study we have been carrying out in Southern Africa, where Rift Valley fever occurs. And on this graphic, you can see the blue bars are the daily rainfall measurements. And then the colored lines represent the different emergence of vectors collected in this Dumbo cold meadows. So the green one is the Aedes species, and the brown one is the Culex. So we basically replicated the artificial flooding experiment under natural flooding conditions. So we know that this theory that was first expanded by Ken Linthicum in 1983-84 actually is repeated somewhere else. Because the models we built were actually based on that artificial experiment in Kenya. But now we have more confidence going forward in terms of building models that this is something that we can apply across the landscape, because we have actually field evidence of the same processes happening in a different region. Now, in linking this to the outbreaks of this particular disease, there are various maps that are shown here, but I would just want to draw your eyes to the 3 bottom maps. And those maps on the right, bottom right, is conditions, rainfall conditions during a La Nina events. In the center, the distribution of the different outbreak patterns of Rift Valley fever across the landscape. And on the bottom left is the rainfall conditions during El Nino events. And what you notice that the areas in green and bluish in Eastern Africa during the El Nino events are typical. And then the drought happening in Southern Africa during the El Nino events. And then the opposites in terms of what is happening. So when you look at the distribution of the outbreak patterns in the center, you can see that they basically reflect the patterns of El Nino and La Nina events over this particular landscape. This is a very interesting finding that we have come up to learn, is because even if you look at many diseases, you don't find this kind of classic correlation between what is happening in terms of the rainfall patterns and disease outbreaks. So is a very good illustration of climate variability impacts on disease outbreaks. So what we have done, we have put together a model that is ingesting these variables of climate, including rainfall, vegetation, and land surface temperature. And then we are running that model on the basis of understanding where there is livestock distribution, human population distribution, and then we run this model over time as shown here on the right. We run the model using a 3 moving window technique, whereby as we get new data, we drop the last information in the model, and then use the new data so that we are able to produce dynamic maps of where the risk will be for this particular disease. The importance of this is that it takes about 60 to 90 days between when it starts raining and when you have an outcome in terms of outbreak. So when you release a risk map every month, it can be used in terms of surveillance and in terms of where to target control efforts. So a good example is this composite map. And I will explain a few things about this map. The areas that are shown in green are areas that we have classified to be potential outbreak areas or potential episodic areas. And then the different dots show you where outbreaks happen between 2006 and 2018. And what we find that using this model that was based on the artificial flooding experiment is that the performance was good in East Africa during the 2006-2007 event, about 70%, 50% in Sudan. In South Africa, 2008 was 30%, but in 2010 to 11, it was 85%. And you'll ask me why the variation in this performance. For example, what we found out in Southern Africa in 2008 is most of the outbreaks happened outside this green area. So the outbreaks were not part of where we were modeling. And then in Sudan, most of the outbreaks happen in irrigated areas, okay? So there is always the whole idea that we have to keep improving. And sometimes if you are trying to characterize where disease occurs, you don't have full information. So being able to build this information over time by observing what is happening in terms of disease outbreak is important in terms of continued improvements in the models. Now the graphics on the right show you the evolution of the disease in the 3 countries, in Kenya, in Tanzania, and in Sudan. The red line shows you when we first provided an early warning of the possibility of an outbreak happening in that area. The blue line shows you when the outbreak started. And what we see is that in Kenya, we had about a period of about 2 months or so between when we first issued the alert and when the outbreak started. In Sudan and Tanzania, we had about 5 months. So if you think about it, that period can be utilized enough to carry out prevention measures, either in terms of livestock vaccination or mosquito control. So from this work, together with the World Health Organization, we came up with this particular model of being able to control Rift Valley fever or any other vector-borne disease that has similar characteristics. So if you think about it, when you utilize forecasting based on satellite information, when you know that the likelihood of above normal rainfall is going to be there during the growing season, what you do is you carry out mass vaccination. If you carry out mass vaccination, you are being able to reduce the number of potential livestock that are affected. If you don't, then you end up in a situation whereby you have this barrage of outbreaks, then hitting the human beings in the end. So the importance of having early warning information is important in terms of controlling the disease. So how is this information important? We often have shared this information with different organizations, and I'm sorry, the text there is misplaced a little bit, I think, when we transfer the presentation to a different platform. We shared the information with USDA, with the Department of Defense. You'll ask me why the Department of Defense. The important thing is that we have many of our men and women deployed in different areas at different risk. For them, having this information will enable them to take proactive measures, either in terms of vaccination, if the vaccination is available, or prevention measures when they are deployed in the field. And then we share the information with USDA in order for them to find out where different ship containers are coming from. So if they are coming from a particular region where the outbreak happened, they are able to place that particular cargo under inspection to minimize the impact. Either vectors are coming in or infected cattle are coming in. We also share the information with the WHO, World Health Organization. This takes care of human health. FAO, it takes care of livestock health, and then an organization called OIE, right now it's called World Organization for Animal Health. This deals with regulating livestock trade products. So they have to know where particular regions are affected by Rift Valley fever in order to reduce the chance of those particular livestock products getting into the supply chain. Now, I would use this graphic here to show the impact of having an early warning system in place. You can see there are 3 pictures in A, B, and C. First, we'll take a look at the picture in C, and this is of a farm near Nairobi, which we have used as a sentinel site for this study. And you can see that in 2006, 2007, even though we provided early warning about 2 months in advance, they didn't vaccinate. Actually, there is, and the uptake of information by administrative systems is often lagging behind, because this was the first time we ever issued an early warning for Rift Valley fever. So they didn't take it seriously. You know, people want to wait. Oh, is there something is going to happen, or is it not going to happen? As a result, on this farm, they lost 80% of their sheep population. Then come to 2016, 2015-16, when you can see on the right, vectors emerge from the particular habitat on this farm. But this time they took seriously the early warning and vaccinated their livestock. And with the livestock vaccination, there was no mortality. So this is just showing you at a small scale, at a farm level, the impact of, the impact of early warning in terms of protecting livestock health. I'm sure that in 2015-16, the farmer was very happy because all the sheep on his farm survived that particular episode. So one of the things we are attempting right now is applying the same methodology to look at the diseases that are happening globally. So currently we are working on models of chikungunya to provide information globally for chikungunya, for dengue, and for Zika. And this is only enabled because we have access to satellite information that enable us to establish metrics in terms of the anomalies happening on the environment, in terms of relating those anomalies to particular disease happenings. Some of this work has actually been featured in a Netflix documentary called Connected. There is a particular episode that is clouds. So if you have access to Netflix, you can go dive deeper into this work. It was shot at Goddard in 2018, 2019, and also in a field study area in Southern Africa. Now, going forward, we have always more data coming in. You can see we started doing this work using the AVHR data. And then we have moved into the MODIS era. The current data we are utilizing right now is MODIS. We have now measurements also from global precipitation measurements and from SMAP that we are now infusing into the models using machine learning techniques. And then in the future, we are going to have more measurements from SAR to map soil moisture, which is very important for particular disease like rainfall fever, and then also the upcoming SBG missions. So more data is going to expand the number of applications we can use in this particular area. Now, one thing just to finish up that I want to point out is nobody has all the answers. My background is in geography, so I have learned by collaborating with people who are experts at disease and people who are experts at disease ecology. Also, collaboration, therefore, is important. It increases the kind of science we do. It provides new avenues to explore. And then also, in order to do this work, you have to persist. If it wasn't for the foundational work of Ken Linthicum in Kenya in 1983, we probably would not have this work coming on board. And then the last thing I would say that climate variability of shifts of whatever flavor matter, either negative anomalies in the climate variables or positive anomalies, as we have seen for Rift Valley fever. All these matter to disease outbreaks, because what we have found is that disease vectors are adapted to different climate conditions and not only one condition. So to finish off, I just want to play for you guys this particular movie. It shows you the evolution of sea surface temperatures and disease outbreaks from 2008, 2009, I believe, until 2014. We'll let it run for a few rounds and I'll point out some things. So what I want you to pay attention to is this period of 2015, 2016, which is during this El Nino of 2015-16. And you see how different clusters and different outbreaks begin to dot the environment. So this work could not have been possible without the collaboration of many people, both from NASA Goddard Space Flight Center, USDA, NOAA, and also Equator Alliance. And we have funding support from different organizations, NASA, NOAA, Department of Defense, and USDA. Thank you very much.
Thank you for interesting talk. I want to ask if there are any pattern between the disease that you showed in the first slide, that was 9 diseases that you selected. Is there any common pattern between them to construct models for that disease?
Yeah, like I mentioned before, before you engage in any construction of a model, you have to understand the biology. And the problem I see right now is that people don't understand the biology and they want to do a model. Before you understand the biology of the vectors, you cannot impose on environmental measurements. So it's very important to understand the biology. That is why collaborating with disease ecologists and people who study the disease is very important to be able to create, I would say, near accurate models. Yeah. So if you pick a disease, you have to find out what vector is driving this disease, and is that vector, can you understand that from the aspect of what is going on in climate? How do populations of that vector, how do the dynamics of that vector population vary in time? Because often, also, even if you have an increase in the number of vectors, there are different aspects of the disease actually we don't understand. For example, if you don't understand the aspect of livestock immunity or population immunity, you may provide an early warning, but an outbreak that doesn't happen. Okay, now you are going to lose your funding because of that. But if you understand that, there is an aspect of immunity that even under perfect conditions, you might not have the realization in terms of a disease outbreak. Of course, the important thing is we don't want disease outbreaks to happen. Okay.
So in some of these areas where, if you immunize your cattle or sheep, how long is the vaccine good for? Why, I mean, is it so expensive that, or it doesn't last long enough? I mean, why wouldn't you just vaccinate all your lambs or,
Yeah, we have been carrying out some studies in Africa to actually look at that particular aspect. I don't have full information because I was not involved in the vaccine study, but I think it's a couple years. The vaccine can be can have an impact for a couple years. But the important thing when it comes to Rift Valley fever, if you have vaccination campaign that is, say, on an annual basis or every 3 years, you'll actually will minimize the impact on livestock. Yeah. The problem is right now there is no livestock for human beings. So even though there are particular efforts right now by an organization called CEPI that is funding particular initiatives to develop human vaccines.
well, we take one for West Nile virus or Lyme disease.
Yeah.
Hey, so I know here you talked about modeling, looking at vectors. I was curious if there's been any research looking at using some of these same variables to model like human wildlife interactions, to look at sort of like how this might impact the crossover events or from like zoonotic jumps from various animals to humans, so far as compared to like just vector-borne diseases.
You mean in terms of looking at crossover?
Yeah, like I didn't know if there were any associations with some of these climatic variables and human wildlife interactions that could kind of drives disease outbreaks?
I know that there is research going on in the area that I'm not very familiar about, but that is a very important aspect in this area. I would say if that is something you are interested in, I think you should go ahead and look at it. It's a growing area, especially when we are looking at this emerging area of One Health. Is important to understand the interaction between the environment and diseases in terms of animals and human populations. So there are people who are looking at those kind of jump over events. Yeah.
Thank you for really interesting talk.
Yeah.
I have 2 questions.
Yes.
And maybe I missed this, but how is the NDVI derived?
The NDVI is derived by looking at measurements in the electromagnetic spectrum, and in the visible portion of the electromagnetic spectrum, there is a low reflectance of radiation in that particular portion of the spectrum from vegetation. In the infrared portion, there is a high reflectance. When you take the difference between the two and normalize it, you end up with an index basically showing you how green the vegetation is on the land surface. Now, if you take those measurements and put them together, like we do using compositing techniques to minimize cloud cover, and you take them over a long period of time, you are basically creating a climatology that you can relate to various aspects of climate parameters, like, for example, rainfall, temperature, and all that. Yeah.
So I was curious on your global map where you showed outbreaks and relationships between El Nino and La Nina.
Yeah.
I think with 2 exceptions, all the diseases listed there were mosquito vectors.
Yes.
The exceptions being cholera and hantavirus.
Yeah.
I was really curious about why hantavirus in the Western US.
Yeah. So
that cluster that responds to
yeah. So the story more
the mouse rodents are
the story there is a
rodents
little
are vectors, right?
is a little complicated, and it's basically during rainfall events there is an increase in rodent populations. Yeah. Rodent populations. So that is related to habitat stability. So if you create conditions whereby you have populations of rodents increasing, you have a tendency to have an increase in the number of hantavirus cases. Yes. And you observe that even in South America. Yeah. But the classic example is really in the Western US.
Thank you so much for this.
Thank you so much.